and welcome to Biology 141, our histology unit. So going in through this section, you have already looked over your cells and different cell processes. You should know your organelles and what those organelles do. So just a quick little review. You have your nucleus starting from the top. That nucleus is going to contain the DNA. The DNA is going to be in a loose format. That loose format is going to be referred to as chromatin. It only is going to condense during cell division, and then it turns into the actual chromosomes is what you think of when you think of DNA. Also inside of the nucleus, you're going to have a nucleolus. Just a minute. And so that's going to be this little structure here. The nucleolus is going to be responsible for making things like ribosomes. Those ribosomes will then exit the nucleus, go through these nuclear pores, and out into the cytoplasm. You will have some free-floating ribosomes in the cytoplasm, which are responsible for helping create proteins, and you'll also find some of those on the rough endoplasmic reticulum, which are going to be here that you see on this blue membranous organelle. The rough endoplasmic reticulum is also going to be responsible for making and putting together different types of proteins that the body will need. You have your smooth endoplasmic reticulum right here. That's going to be responsible for helping to detoxify the body. It's actually able to break down substances such as alcohol, drugs, caffeine, anything that you take into your body. It could be Tylenol. It's going to break it down here at the smooth endoplasmic reticulum and detoxify it and keep it from harming you. If you've built up a tolerance to these types of medicines, it could be simply because you have built up a large number of smooth ER. So they're able to take these substances apart very quickly, so it requires more in order to get the same outcome from those medications. Moving on over, you have the flagellum here. With the flagellum, that is only going to be found on male reproductive cells, which are going to be the sperm in animals. Coming on over, you have lysosomes. Lysosomes are going to be responsible for holding digestive enzymes. These digestive enzymes are going to be able to break down anything from old, worn-out cell parts to microorganisms that might get into the body. The next, you see a centriole. The centrioles are responsible for creating spindle fiber. These are going to help the cell during mitosis, and they also create some spindle fibers and cytoskeleton fragments that can help things move throughout the cell. But the centrioles mainly help with mitosis and cell division there. Next, you have the peroxisome. With that peroxisome, it's going to contain a type of peroxide that your body actually makes, and that's going to help break down things such as alcohol substances. Coming on down to the cytoskeleton, that cytoskeleton is going to be made up of 3 different types of filaments. You have what's referred to as the microtubule, you have the intermediate filament, and you have the microfilament. All of those are going to make up the cell cytoskeleton, and they're going to help move things around the body or around the cell, the body of the cell. Coming on over to the Golgi apparatus. Once proteins and substances are created within the body, or the body of the cell, then they're going to be sent to the Golgi apparatus. At the Golgi apparatus, they are going to be activated and then packaged for export. Now that export could be to other parts of the cell, or it could be to other parts of the body. When they get packaged for export, they're going to create these little circles here, so basically put its own little cell membrane around it, or a vacuole around it, and those are referred to as Golgi bodies. Next, you have the organelle that all people know that is your powerhouse of the cell. That's your mitochondria. Notice how it looks like a kidney bean shape, and it's often referred to as a sac within a sac because it has 2 membranous sacs in this organelle. This is where your food is going to go, and it's going to be broken down and made into actual energy molecules that you can use, and those are going to be your ATP molecules. All right, coming back to the smooth ER, I forgot to mention that they don't only break things down, but they're also responsible for creating things within the body, such as different lipids that you might need, like cell membranes. They also help create different steroids that you use in your body, such as testosterone, progesterone, and estrogen. Right, this is a close-up look of the cell membrane. With the cell membrane here, you're going to notice that you have your hydrophilic heads. These are actually going to be made up of the phosphate or glycerol heads. You're going to have your fatty acid tails. They're going to be at the bottom part. They are actually hydrophobic. You have fibers that are going to be there within that extracellular matrix. You have proteins throughout the cell membrane. This one is actually going to be referred to as a glycoprotein. The purple part is the protein part. The top part is the carbohydrate, which is the glycogen. Those can help with cell recognition, so the body knows that those cells do belong there. Within, embedded throughout, you're going to see these cholesterols. These cholesterols are going to be responsible for keeping that cell membrane fluid. However, if it becomes too hot, they can become rigid and become distorted. So what exactly is tissue? Well, it's going to be a collection of similar cells that work together within the body to perform a specific function. So how's tissue exactly going to be made? Different tissues are going to start out from a stem cell, and from those stem cells, they're then going to differentiate depending on what part of the body that they're in into the tissue that that part needs. So it creates those cells, and then it puts them together into a functional unit. These cells need to be able to stick together, so we're going to talk about different types of cell junctions. So cell junctions are going to be there to hold these cells in a number of ways. They're going to be points of contacts between cells where they're able to either send chemical signals through. Our next type of junction is referred to as a tight junction. These are going to fuse cell membranes between cells together. They tightly join them through a transmembrane protein. And this is going to be important because this is going to be found in things that you don't want to leak, such as the intestines and the stomach and the bladder. You want these things to be waterproof and tight, so they keep everything in. Looking at tight junctions, this is what they would look like between two cell membranes. You see how that you're going to have that space in between, so you have an intracellular space, and you're going to have those little strands of transmembrane proteins that are holding them together. So they look like little balls that are holding the two cells together. Adherence junctions are going to form belts of adhesion. These are going to be used with plaques with similar transmembrane proteins that are stuck to that plaque. And so we'll look at a picture of that in just a minute. So the plaques are going to act like Velcro, and they're going to hook these cells together, but allow some flexibility. So you're going to have one end connecting with the other, kind of like Velcro does. You'll find these in things like the esophagus and the small intestine, things that need to be able to stretch but still hold together. So looking here, you see that microfilament actin going through that plaque, and that's going to create an adhesion belt. And then within that, you have the transmembrane glycoproteins called cadherins that are acting like the two hands that are holding together, like in Velcro. Desmosomes are going to be good in keeping things together. They are going to act like spot welds or cell rivets. They're there with their protein plaques as well that are anchored in by the cytoskeleton. So you'll see cytoskeleton filaments that are attached to it. The attached to plaques are transmembrane glycoproteins, or the cadherins, and these are going to act like zippers that kind of zip the cells together. These also are going to give flexibility, but they're going to be very strong, and they're not going to allow that tissue to tear apart or to be separated, those cells from each other. You'll find these in things like skin and heart muscle. So looking here, you see the two plasma membranes. Of course, they have that intercellular space, which is going to separate them. You'll see the plaque with the cytoskeleton attached in using the little blue particles, and then you'll see the transmembrane glycoproteins also hooking them together. But this time it's going to act more as a zipper. Next, you have what's referred to as a hemidesmosome. Hemi, of course, means half, so that's technically just half of a desmosome. These are going to have their transmembrane glycoproteins. They're attached to plaques. They're anchored to the cell basement membrane. So that's going to be the bottom part. They're going to keep skin in place. So this is what anchors that bottom layer of skin in. And looking at those, you can see you have your intermediate filaments, which are going to be part of that cytoskeleton. They are made of keratin. You have the actual transmembrane glycoproteins there, which are holding them down to that basement membrane. And then you see that plaque that they're attached to. So that's a hemidesmosome, which is half of a desmosome. Next, you'll have gap junctions. These are made of proteins called connexons. They're going to allow molecules to move back and forth between cells. It can allow for chemical communication between these cells or electrical signals to be sent through. They're going to be found in things like embryonic cells, and you'll see these in, for example, heart muscle cells used to transmit electrical signals through the pores. They can send chemical signals to let the cells know that there is a virus or that there is a other type of molecule that's in. So they just transmit chemical signals back and forth. And so looking at gap junctions, you'll see them here. You have, of course, your two cell membranes again, and you'll see those connexons that are attaching the two. And they act like channels or tunnels that allow signals to go back and forth. And so here are going to be all of those. You will need to know all 5 of these. The 4 tissue types that are in histology are going to be your epithelial, your connective, your muscular, and your nervous. And with the nervous tissue, that's going to be responsible for internal communication. So it's going to consist of your brain, your spinal cord, and your nerves. With muscular tissue, this is going to contract to cause movement, and we're going to talk about 3 different types of muscular tissue. So you'll have the skeletal muscle, which is what you usually think of when you think of muscular tissue. That's going to attach to the bone and move you around places. You'll have the muscles of the heart, which are going to be your cardiac muscles. And then you're going to have your muscles of the hollow organs, and that's going to be the smooth muscle. Epithelial tissue is going to form boundaries between different environments, so it's there to give a boundary, to protect. It secretes, absorbs, and it filters. So some examples of those are going to be skin on the surface, which is referred to as your epidermis, and the lining of your GI tract organs and other hollow organs. And the last type of tissue is connective tissue. That's there to support, protect, and bind other tissues together. So that'll be things that you might not even think of as a connective tissue, such as bone or blood, but it's also going to be tendons, ligaments, and fat and other soft padding in tissues. So starting out with the epithelial tissue. Characteristics of the epithelial tissue are going to be cells that show polarity. So you'll have slightly negative and slightly positive ends.
Here's what I found.
It'll have an apical or free surface, which can have a smooth surface. It could have microvilli on it, or it could have cilia on it. You'll have a basal surface, which is going to be the bottom part. It's attached at that section. It'll sit on a basement membrane. They are avascular, which means that they do not have a vascular system, so they don't have blood vessels running through them. However, they are going to be close enough to where they can get nutrients in and get rid of waste products. So that will work through diffusion. Cells are going to be rapidly and frequently dividing through mitosis. Cells are going to be closely packed together. And they're going to form sheets held together by the junctions that we just looked at. Also, they are highly regenerative. Even though they don't have direct supply or a vascular system of blood coming in to help them repair, they are close enough that these things can diffuse across so they can repair themselves very quickly. And they're supported by other connective tissues. So looking at these surfaces of epithelial cells and basement membranes, of course, the epithelium is going to be the cells that you see there. With red. So this is your epithelium. That's going to be that entire cell through here. The top part is the apical part. Like we said, it could be smooth. You might see microvilli on it, or you could see cilia on it. The bottom part is going to be your basement membrane, and it actually is made up of 2 layers. You have what's referred to as the basal lamina, which is going to be that first part, and then the bottom part is going to be the reticular lamina. And so between the two, those both make up that basement membrane. And then you're going to have the other connective tissue that it's being connected onto at the bottom. And that's what is actually going to hold that tissue in place between that basement membrane, the basal layer, and that connective tissue. So with the basal lamina, like we said, it's adjacent to the basal surface of the epithelium. It's a thin supporting sheet. It's non-cellular adhesive sheet, so it's not actually made up of specific cells. It's just a substance of non-cellular adhesive material. It consists mainly of glycoproteins that are secreted by epithelial cells and some collagen fibers. It's a selective filter for molecules that diffuse into the epithelium. And it's going to act as scaffolding where the epithelial cells can migrate to repair a wound. Epithelial tissue functions. It's there for physical protection, so to protect from anything that might damage what's inside. It controls permeability, so it controls what can come in and what can exit. It does this through membrane proteins. It provides sensation, so you'll have free nerve endings there that'll pick up on hot, cold, and pain, but you'll also have some deeper nerves inside which pick up on pressure. It produces specialized secretions from the glandular epithelia, so you might have things such as sweat or such as sebum, which is going to be oil produced. And they're classified with specific cell arrangements. So when you're naming these cells, you're going to have to give me two things. I need to know how many, and I need to know what type of cell. So the arrangement of layers, for example, you have simple, you have pseudostratified, and stratified. With the cell shape, we're going to have squamous, cuboidal, and columnar. And we're going to talk about each of these. Now, when looking at these and naming them, you can see which ones can go together with this chart. So you'll need to make sure that you know which ones go together. We have simple, and so with simple, you can have one layer, and that can be squamous, cuboidal, or columnar. So simple means one layer. Squamous is going to be flat. Think of squished. Cuboidal, of course, is cube-shaped, and columnar is column-shaped. Pseudo-stratified, pseudo means false, stratified means many layers. So this looks like many layers, but it's not. It's only one layer. And we'll look at this in a minute, and it's going to be how they are arranged and how it looks like with the different nuclei inside. So with these, they can only be columnar if you look at the chart. Going down to stratified, that means many layers, and you can have this with the squamous, the cuboidal, or the columnar. So epithelial tissue, we have covering and lining epithelium. That's going to be external and internal surfaces of the body. We have what's referred to as glandular epithelium. That's going to be the secretory tissue as part of glands, so it's going to secrete things. First up, we're going to look at simple squamous epithelium. So with the simple squamous, you have one layer that you see up there, and it's going to be a flattened cell. Think of a squished cell. So that's your simple squamous. Single flattened layer. This is going to be important in places where things need to be able to fuse across a membrane very, very quickly, or in and out of a cell very, very quickly. So that'll be things such as your respiratory tract, especially in the alveoli inside of the lungs. The blood and lymph vessels, you're going to need to be able to send nutrients and oxygen into the tissues around, but you're also going to need to be able to pick up carbon dioxide and waste products. Also, if there's some type of injury, it's going to have to be able to send blood particles such as platelets or white blood cells to that area very quickly. So it needs to be very fine and thin, and we're going to find that with our simple squamous epithelium. Also, you'll find it in some membranes that line body cavities. This is what it looks like. One layer, flat, has a basement membrane, has its connective tissue. Over to the side here, this in this area, sorry, it didn't draw correctly, but that is going to be your blood vessel, and you see that red blood cell there just to show you how close these blood vessels are, but they don't actually go into the cells, so those cells are considered avascular. Moving on, we have what's referred to as our simple cuboidal cells. With the simple cuboidal cells, they are a cube shape, simple, which means they're going to have one layer. And they usually have an opening, which is going to be the lumen. That lumen is where things are going to be secreted into it. So it's going to function in forming things like glands, which secrete, and kidneys, which are going to absorb. They're also going to be found in things like salivary glands, kidney, pancreas, and liver. So looking at it, you see your simple, one layer, cube-shaped cells, has the basement membrane, you see the connective tissue there, and you see the blood supply over to the side. Next, we're going to have our simple columnar epithelium. With these, it's going to be one layer of elongated cells. You'll see the nuclei are going to be located near to the basement membrane. Now, you will see some cells there within that do not have the microvilli on top, or it could be cilia on top. You see them down towards the openings there. Those are going to be goblet cells, and those goblet cells are going to be responsible for making things like mucus. Their function is in movement of mucus and oocytes. So the oocyte's what we refer to as an egg. So the proper name is oocyte. It's also there for protection and secretion of digestive fluids and absorption of food in small intestine and microvilli. So I can move that down, get that out of the picture so we can see. This tissue, like we said, also contains those goblet sites, which are going to create mucus. So here in this picture, you're going to see simple columnar cells. Those goblet cells are creating mucus, which you see rising to the top through their little vesicles. You see the microvilli, which are going to be very important. Microvilli increase surface area. The more surface area that cell has, the greater the ability it has to absorb things or secrete things. So it's really going to help with the intestines. Within each one of those columnar cells, notice how the cell membrane is going to be located closer towards the bottom of that cell, and it's going to have a basement membrane. You see your connective tissue, and you see that blood supply source. This one is going to be simple ciliated columnar. Notice how instead of having the microvilli or finger-like projections, it's going to have cilia. So the cilia is going to be responsible for working in a wave-like motion in order to move things across it. It could be particles that mucus picked up from breathing, or it could be moving an egg through the uterine tubes. With our pseudo-stratified columnar cells, remember pseudo means false, so these look like multiple layers of cells simply because of the placement of their nuclei within the cell and how sometimes part of that cell can be kind of smushed to the bottom and you might not see it going all the way to the top. But these technically are only going to be one layer. Commonly, they contain cilia and goblet cells to produce mucus. These are going to line things like the airways, helps keep the airways clean in the lungs. And looking at these here, you can move this out the way. You can see these pseudo-stratified columnar cells. You have your goblet cells creating the mucus. You see the cilia where that mucus is going to be applied. They'll help capture things in the airway and help you cough them out or sneeze them out. Transitional epithelium is going to be epithelium that's able to change their shape and size according to how much they're being stretched. So it's going to be many layers of epithelium, so it is stratified. They can change shape based on the stress, like we said, that's placed upon them, and they're going to be found in things like the urinary bladder, which allows it to change shape as it fills up. So they might be nice, plump, thick cells, and as it fills with urine, they're going to stretch out and they're going to become thinner, looking like squamous tissue. So looking at these are going to be nice, plump cells. You have an apical surface. You have the transitional epithelium, which is going to be the stratified cells or the many layers of cells there. You still have your basement membrane and you still have that connective tissue. Up top, you see those nice and plump. At the bottom, you see them as they're being stretched out. Next type that we see here is going to be a stratified squamous epithelium. With these, you're going to find many layers of these flattened cells. Now, at the bottom, you see all of them do have active nuclei, but as it goes towards the top, some of those nuclei start to die out and they start to flatten out. These are going to be found on outside coverings, usually things such as the skin, or you could also find them in the wet skin on the inside, so dry skin and wet skin. If it's going to be dry skin on the outside, they're replaced by a cell referred to as a keratinocyte. They fill up with the protein keratin, which becomes waterproof for these cells. So that nuclei starts to die out, and then those are also going to be the cells that you slough off through the day or shed through the day. Like we said, they occur in both dry places and wet places. So the skin on the outside is the dry. That's going to be the keratinized stratified squamous epithelium, and then the ones inside, which are found in things like inside of the mouth, the pharynx, the vagina, the throat, anal canals, those are going to be wet types, and so you won't find that keratin substance there, which is going to help keep them dry, and so those are non-keratinized stratified squamous epithelium. And it's going to also form a protective layer for these areas. You can't see that at the bottom. Right, so looking here, this is just an artist's rendition. You see the flattened squamous cells on top. Notice that those have died out. The ones underneath are going to start to flatten, and then as you go towards the bottom, they are going to be thicker and more plump. They're attached to a basement membrane, and you have your connective tissue layer there. These are looking at the skin on the outside of the body. Notice how the ones on top are going to pretty much be the dead surface cells. These are the ones that you slough off through the day or you shed through the day. The ones underneath, notice how they still have nuclei, so they are still living. Right, our next up is going to be stratified cuboidal epithelium. In order to be stratified, it only has to have 2 or more layers. And then those layers in this case are going to be cube shaped. So they consist of several layers of cuboidal. Then in the inside you will have that opening still, and that opening is always referred to as a lumen. It's going to perform or give it a protective tubing there. It's going to be found in things like your mammary glands, which produce milk, sweat glands, which produce sweat, salivary glands for saliva, pancreatic glands, and the urethra. And so here is what they look like. You have your apical surface, you have your basement membrane, so it'll have a basal surface attaching to it, connective tissue, and then you see your blood supply over to the side. Stratified columnar epithelium consists of several layers of column-shaped cells. The function is going to be there for protection and secretion. It's found in places like the male urethra, the vas deferens, the pharynx of males and females, the anus, and the conjunctiva of the eye. So looking here, you have your stratified columnar, which just needs to be 2 or more layers. You see the apical surface that has a basement membrane, connective tissue on the bottom. So, tissue levels of organization. What structure is being pointed at in the images below? What do you think you're looking at? That is going to be that basement membrane. Glandular epithelium is going to be composed of cells that function to secrete substances. This is going to help to form things like glands. 2 classifications of glands based on where they secrete their products. Exocrine glands secrete their products into the external or internal surfaces. Endocrine glands are going to secrete their products directly into the bloodstream or bodily fluids. There's going to be 3 types of exocrine glands that you need to know. We have merocrine glands. They secrete a watery substance. Apocrine glands, which secrete portions of a cell. And then holocrine, which are going to secrete whole sections of that cell or a whole cell. So looking at these, you have your merocrine, which you're going to find there in the pancreas underneath the stomach there. And so it's actually secreting its secretions into the opening or that gland section. Next, you have the apocrine. There we see it creating milk, so it's actually breaking off part of that cell and creating that milk and secreting it into the gland. And then you have the holocrine glands. With those, you see the actual whole cell breaking off and becoming part of that substance. And in this case, in this last picture, is going to be sebum, or I lost it. Sebum, or it's going to be referred to as the skin on your, or the oil on your skin. Sorry. So picking up next time, we're going to pick up with connective tissues.